In this lecture, we will explore what life needs to develop and how life likely developed on Earth. Earth has had three atmospheres. The early atmosphere, or first atmosphere, was an accretionary atmosphere of hydrogen and helium. These are light gases, and they escaped owing to low gravitational attraction and were blasted away by the solar wind. Earth's second atmosphere was still not the same as what we are used to today. In fact, we would not have been able to survive in atmosphere 2. Atmosphere 2 was created by differentiation of the Earth coupled with solar UV radiation. Because of differentiation, the early Earth was very volcanic and many gases were being expelled into the atmosphere. These volcanic gases included water vapor, carbon dioxide, hydrogen sulfide, and nitrogen. From these volcanic gases and UV radiation from the sun, small amounts of other gases were created. These included ammonia, methane, carbon monoxide, ethane, formaldehyde, and importantly, hydrogen cyanide, which is a critical building block for amino acids and nucleic acids. Life began in what we would consider a most inhospitable environment. Meteorites were transporting water and organic molecules to a primitive ocean. The ocean and atmosphere were bathed in UV radiation because we had no ozone layer yet. And electrical discharges in the form of lightning were taking place constantly. It was a hellish place, and it is in this environment that life began. So let's step back for a second and ask the question, what is life? Life exists in cells, and to be considered a living thing, an organism must contain the following 5 properties. They must have an organized structure. They must be able to self-replicate, have some form of information storage, have behavior, and have energy flow, known as metabolism. Cells are basically chemical factories. Life depends upon chemical reactions, and these reactions must be isolated from the outside environment or they would not work. The cell, like this simple prokaryote illustrated, provides a protected environment for these chemical reactions to take place in. The first step toward life on Earth was chemical evolution. Chemical evolution is the mechanism by which lifeless organic compounds developed the characteristics of life. Although this process is not yet recognized in the rock record, at some point in time, the organic soup changed from chemical evolution to biological evolution. In 1953, Stanley Miller exposed a mixture of gases to an electric discharge. Chemical reactions ensued that produced formaldehyde, hydrogen cyanide, and amino acids, the building blocks of nucleic acids, proteins, and life. This experiment has been repeated countless times with similar results. The organic soup that may have been the precursor to biological evolution was produced. In fact, the amino acids formed in the experiment are the most common in living cells today. This is a diagram of the apparatus that Stanley Miller used in his 1953 experiment. The oceanic compartments and the atmospheric compartments were filled with water and gases that were set to mimic the early ocean and Earth's second atmosphere. The electrodes were introduced to simulate lightning. Amino acids grew on the electrodes, and the water quickly changed color due to organic molecules that grew when the condensed gases were introduced. The one gas that is common today that we need to survive is absent from Miller's atmosphere compartment. That is because oxygen is destructive to amino acids. A soup of organic molecules, however, is not life. So, how did life develop? Organic molecules need to be concentrated to stimulate reaction to form proteins. Concentration mechanisms can include things like evaporation, freezing, enclosure inside bubbles and drops, and absorption into minerals. Evaporation reduces the water content in, say, a pond and crowds molecules closer together. Freezing does the same thing by turning water to ice and concentrating organic molecules in a confined space. Each of these mechanisms may have played a role at some point in time. Now, we have established that life requires amino acids, but it also requires nucleic acids known as RNA and DNA. Nucleic acids are structures made of base pairs called nucleobases that consist of sugars and phosphates. Sugars and phosphates link to form nucleotides, which are fragments of DNA and RNA. Long nucleotides form very easily on phosphate minerals and clays. Conditions simulating Earth processes have been utilized in the laboratory production of sugars. These simulations include waters from hot springs flowing over clay beds and reactions involving electrical discharges to simulate lightning. Phosphates are also associated with volcanic activity. So the conditions necessary for the precursors of life existed on early Earth. Still, amino acids and nucleic acids are not life. In 1969, though, a meteorite fell to Earth in Australia. It was named the Murchison meteorite. Fatty acids extracted from the Murchison meteorite formed lipids when they were dried and re-wetted. In a laboratory setting, shaken mixtures of lipids, amino acids, and nucleic acids will spontaneously form vesicles. These vesicles trapped organic molecules, basically forming tiny reaction chambers. So it is only when we combine lipids plus amino acids plus nucleic acids that we can get the right components for the chemical factories needed for life. So the next question is, where did life evolve? Cold climate volcanic islands have preferred conditions. They have fresh or brackish water that is necessary because organic molecules are destroyed by salt. These islands often have lakes or river-fed lagoons. They have low temperatures, which are important because RNA becomes unstable at temperatures greater than approximately 25 degrees Celsius. Cyanide and formaldehyde reactions needed to form organic molecules form very easily in half-frozen mixtures. Lastly, volcanic eruptions generate lightning, which is energy, and provide clays for amino acids to grow on. This is Surtsey. Surtsey is a volcanic island that erupted out of the North Atlantic Ocean off the coast of Iceland in 1963. It is a scientific investigation site where the questions of how life can develop on volcanic islands in cold climates is being investigated. So what about black smokers? Black smokers are deep marine hydrothermal vents that are associated with divergent plate boundaries. They support a vast array of life forms that do not require photosynthesis. Cold, dense seawater sinking into the fractured ocean floor crust basically encounters hot rocks heated from underlying magma. The hot water is chemically aggressive and dissolving large amounts of metals from the surrounding ocean crust. The hot water, as hot as 500 degrees Celsius, is less dense than the surrounding materials and rises to the surface, where it erupts as a geyser of hot water on the sea floor. But because the water temperature on the sea floor is around 0 degrees Celsius, the erupting hydrothermal water rapidly cools and becomes supersaturated with the dissolved minerals, which precipitate as tiny grains of metallic sulfides, creating a black smoke. The elements needed for life to evolve exist here in a very different environment. All cells require energy because energy drives chemical reactions. The most abundant and readily available source of energy is solar energy in all of its manifestations, such as heat and light, wind and ocean currents, and fossil fuels from decayed plant life. Metabolism, energy, is simply electron transport. Where we get those electrons and where we put those electrons defines the type of metabolism we have. So heterotrophy is life's first energy. It is energy gained from the breakdown of organic compounds, in a sense, from eating things. Electrons come from organic molecules or the food molecule, and they pass to a terminal electron acceptor. Aerobic organisms use oxygen as their terminal electron acceptor, while anaerobic organisms use other molecules. Anaerobic organisms usually break down sugars through fermentation in the absence of oxygen. Autotrophy, or self-feeding, is self-generation of energy by getting electrons from inorganic compounds. Chemosynthesis, also called lithotrophy, uses inorganic compounds to build organic compounds. For example, methanogens belong to a unique group of organisms called archaea. Methanogens break carbon dioxide bonds and transfer carbon to hydrogen to build methane and transfer oxygen to hydrogen to yield water and methane. Excess electrons that are used for energy for the cells. Photosynthesis or phototrophy is feeding by light. Certain organisms contain a molecule called chlorophyll that becomes activated by specific wavelengths of light. Activation of chlorophyll produces electrons, which are used in reactions between inorganic compounds that yield organic compounds and energy. The first photosynthetic reactions probably used hydrogen or hydrogen sulfide. Now, photosynthetic bacteria eventually switched to very abundant water as their hydrogen source. The problem here is that monoatomic oxygen is very toxic. It is a deadly poison because it oxidizes organic molecules. This problem was solved by the cyanobacteria, which produced a critical antioxidant enzyme called superoxide dismutase. This enzyme combined monoatomic oxygen to make O2, which is less harmful and can be expelled easily through the cell wall. After the arrival of photosynthesis and the production of O2, our modern atmosphere, atmosphere 3, begins to evolve, and it evolves over vast stretches of time. Now, although there is no apparent geologic record, one may hypothesize that the appearance of oxygen in a world where life evolved without oxygen would have caused a significant extinction event. Could this have been the first great mass extinction event? In summary, early Earth had a large biological energy budget. There was a large population of microorganisms that include archaea, photosynthetic bacteria, and heterotrophic bacteria. This is the end of Module 4. In Module 5, we will begin our travels through Earth history and investigate Earth's earliest life.